Glycogenesis is the process by which glucose is converted into glycogen for energy storage in the body. Glycogen is commonly stored in the liver for blood glucose control and stored in the skeletal muscle for a quick energy source. Once glucose enters a cell, it must complete a few reactions in order to become glycogen, starting with the phosphorylation of glucose, creating glucose 6-phosphate. This reaction is catalyzed by either hexokinase or glucokinase using ATP, depending on where in the body the phosphorylation takes place. The muscle cells use hexokinase, which functions efficiently during fasting times due to a low Km. Hexokinase is not induced by insulin. Glucokinase catalyzes glucose phosphorylation in the liver, and it is induced by insulin. Another difference between the two is that hexokinase is allosterically inhibited by glucose 6-phosphate, while glucokinase is not. The phosphate group on the 6 carbon of the glucose 6-phosphate is then transferred to the 1 carbon by the enzyme phosphoglucomutase, forming glucose 1-phosphate. Uridine triphosphate then reacts with the glucose 1-phosphate to activate it. Uridine monophosphate from the UTP is coupled with the glucose 1-phosphate to form uridine diphosphate glucose, UDP glucose. The UDP glucose is then catalyzed by glycogen synthase and attached to preformed glycogen primer and creates an unbranched chain of glycogen. Glycogen synthase, an insulin-dependent enzyme, does not have the capability of forming branching points on the glycogen. Through these 4 steps of phosphorylation, phosphate transfer, UMP coupling, and glycogen synthesis, a glucose molecule is added to the ever-growing glycogen chains used for energy storage in the body.